biomarkers for lung cancer metastasis. We use a cell model to find out the protein candidates, and then verify their validity using clinical specimen. Yeah. The reason I'm interested in lung cancer metastasis is because in Taiwan, cancer is the leading cause of death, and lung cancer is the leading cause of cancer. So the Taiwan government actually formed a big research program to get scientists to work on the lung cancer problem. So I started about 10 years ago participating, we call the National Research Program. So one subgroup, many scientists are working on the lung cancer in Taiwan. The big challenge for the lung cancer problem is if we can understand the mechanism of metastasis, then you can get a much better survival rate. Here's a number. If you can detect the cancer in the early stage, you just do a surgery and remove the cancer, and maybe use some chemotherapy. Most of them, they are cured. But if the tumor metastasized, then you have trouble. Usually the survival rate become very low, lower than 10% or even much lower. This figure shows you about how people think about the process of metastasis. So first, the cancer cell has to leave from the primary site. That means they need to get loose. Maybe it secretes some protease to dissolve the extracellular matrix, and then they will get into the circulation system and then get out and try to land to the distant site, and then form the new tumor. So as far as we understand, cancer metastasis is at least partially related to the secreted proteins, just like what I described. If the cancer cell can secrete a protease or other enzyme activity, then there's one way to change the extracellular matrix. Or sometimes they have proteins on the membrane surface, so they can bind to the extracellular matrix. All of them, they were somehow related to metastasis. So if you look at the literature, you will find some, like galectin-3, is a secreted protein, is related to the metastasis. Some drug target, they are also related to secreted proteins. And some biomarker, for example, we published a paper to find out the secreted protein from the pleural effusion. Eventually, we demonstrate is related to the prognosis of the distant metastasis. So we become interested in looking for the secreted protein from the cancer cell. Now we have a picture in mind just like this. If we try to understand the secreted proteins, the collection of them we call the secretome, we can use the proteomics methods, especially mass spectrometry, because I'm familiar with the mass spectrometers. Then we can define cancer-related secreted proteins. Somehow they are related to the disease mechanism. Since they are secreted protein, eventually they will go into the circulation system. So it's very easy to develop assay and use them as a biomarker to do diagnosis. And also, they could be the therapeutic target. So what we did a few years ago is we proposed a project to the National Research Program. We hypothesized there should be many more secreted proteins. We just don't know where they are, but the proteomics methods will help us to find them. So we try to work on a cell model. In Taiwan, the scientists, they culture the lung cancer cells with different metastatic power from the male patient. So we decided to use this cell line for the cell migration and invasion functions. So that's the cell model. It just like I just described. Actually, they obtain the cell from a male lung cancer patient, and then they use the transwell device and select subpopulation of the cell repeatedly, and then they get a highly metastatic subpopulation. So what we can do is we can compare the secreted proteins from these two cell lines and try to find the potential target from the difference. But before we decide to do so, we need a strong hypothesis and make us believe there should be something there. Then we can really invest our experiment in this experimental design. So we demonstrate one evidence. If you acquire the condition median from the CL1-5, the highly metastatic cells, and apply this condition median into the less metastatic cell line CL1-0, you will find these cells become more migrative and more invasive. If you do the opposite way, the condition median from CL1-0, they somehow inhibit a little bit about the cell migration and the invasion. So, which means that there must be something from the condition median from these cells, but we just don't know what they are. So it's a typical project idea for the proteomics research. But when we try to decide to work on the secreted proteins, we realize it's not that easy because especially when you work on the mammalian cells, you are facing at least three problems. First, the secreted protein, the amount of secreted protein could be low. And most of the mammalian cells, they are cultured in the cereal containing medium, and they could be interferencing for the proteomics analysis. The most troublesome is the third one. If you culture the cell in a dish, most of the time the scientists will say, well, they are viable, they are happy, and describe the viability. Usually it's in terms of 99% or 98%. But how about this 1 or 2%? If they lyse, they release all the proteins into the condition median, then they are in trouble because the biomarkers you are interested in, they are in low abundance. So it causes another problem. So at first, we try to follow the protocol published in the literature. You will find the so-called secretome analysis displayed on the SDS page gel, almost identical with the cell lysate. You can just lyse the cell and look at the pattern and compare to what you will find in the literature. They are pretty much the same. So people are trying to find a small amount of secreted proteins within very abundant proteins from the lyse cells. So after different attempts. So basically, my graduate student and I, we just try different systems, see if we can get a better result. Eventually, we found this system is pretty good for our purpose. It's a hollow fiber culture system. Basically, they are commercially available now. They just bundle a lot of hollow fibers, and this cartridge into a cartridge, you can grow the cells between the fibers, and these fibers will be like a capillary in the body. So with a pump, you can pump the medium through the hollow fibers. Because these hollow fibers, they are permeable, so you supply the nutrient and oxygen, those things, and then remove the waste from the cells. So that's pretty much very physiologically comparable to the living organism. So there are 2,800 fibers in one cartridge. The fiber is very small. It's only 180 micron. And so the surface area is huge. And they grow into three-dimensional. They are not just, they don't just attach to the surface. They just grow into the three-dimensional culture. So you can get a huge cell number. It's comparable to more than 100 dish for a single small cartridge. So we use the 5 kilodalton cutoff hollow fiber. You can choose different cutoff. That means if you will get a concentrated secreted proteins in the extra capillary space. That's what they call it. Okay, and you can collect them. Sure, you will not get the small molecules if they are less than 5 kilodalton. But using this system, you can you will end after a few days. You will end. For example, we collect the protein every day. You will end up with a concentrated protein in a very small volume, less than 5 ml. And one thing we did not expect, but it actually helped a lot, is we found the cell lysis rate is extremely low in this system. We did a comparison. What we did is we used these housekeeping proteins. They don't secrete it into the outside the cell. That's what we learned from the literature. So we can use it as a marker to see how much protein in the conditioned medium, and then we just lyse a known number of cells and do a calibration. We can actually calculate the cell lysis rate. So for the red ones, they are traditional dish cultures, and these gray ones, they are hollow fiber culture system. You will find, as most people describe in the literature, using a dish, you will get like a 1% cell lysis rate, which translates into a 99% viability. But for the hollow fiber system, you get a very low lysis rate. I think that just because they are growing, these cells grow into the three-dimensional structure, is much comparable to physiological condition. That's why they are happy. They don't just lyse due to some unknown reasons. So we use this system to culture some cancer cells. The first example we did is we did on the nasopharyngeal carcinoma. This is just a purely descriptive study. So we just culture the cell and collect the protein, and we found a small database with a few hundred proteins. And then we go to the literature, try to find out what these proteins are. Most of them they are reasonable, but some of them, like this chloride ion channel 1, it's kind of you suspect that it shouldn't be there. Then we just purchase the antibody or raise the antibody and try to compare the cancerous tissue and adjacent normal. And you will find they are in huge, the protein abundance in huge difference. And then you realize you might find a potential biomarker target. For the CLIC1, you can compare the plasma concentration using a ELISA kit for healthy controls and different cancer type. You'll find this protein is especially the concentration is especially high in the NPC patients. And you can also work out the RC curve to see if it's good for the diagnosis. And you can compare the plasma specimen from patient in different cancer stage. For this one, the concentration is raised at the early stage. So we publish this result and consider it's early stage, early diagnostic marker. Okay, let me get back to the cell model. So I already described the CL-0 and -1 and -5. They are lung cancer cells with very different metastatic power. So what we did is we just used a system to collect the secreted proteins and do the label-free mass spectrometry-based proteomics analysis. And then we can just do a statistical analysis to find out which one has differential label between two cell lines. To do this, we developed a simple technique. Since secreted protein is not like what Steve described, to work on the protein phosphorylation on the whole cell, so you need to collect a large amount of sample, like tens of milligrams, and then do a fractionation, and then do a very comprehensive proteomic analysis. What we did is, we know it's a secreted protein. It can be very complicated. It could be, but you would think it's much simpler than the cell lysate. So what we did is we developed a technique called staking gel aided purification. So what we did is we just get a conditioned medium and concentrate them down, and then put on the SDS page gel, but with a staking gel on the top, just regular SDS page. But when you start the electrophoresis, these proteins will focus on the interface, right? Because when they hit a high concentration SDS page, page gel, they slow down. They have to line up somewhere. So all you need is just cut off the small piece of gel and do the standardized in-gel digestion. That's very important because if you just work on the conditioned medium, you need to work on the digestion, optimize the digestion protocol almost every time. But if you do the in-gel digestion, they kind of normalize all the condition, and it's easy to work with. And you can, what we did, my graduate students, they don't even work on, they don't even need to do the digestion by themselves. They just send it to the technician, because the technician in the lab, they do many in-gel digestion every day. So that's the result we have. You end up with a list with some proteins. They have a higher level in the more metastatic cells, and that's the others have with the higher level in the less metastatic cell. And we compare the list with the literature. List with the triangle, the red one, that means they are reported in the literature, they are related to the lung cancer metastasis, like fibronectin. And if it's pink, they are related to the cancer metastasis, but not lung cancer. So as you can expect, a lot of protein they are already is reported in the literature. But one thing I still don't fully understand is you see more triangle on the top, not the other side. I think there's a bias of a human hypothesis. So when people think about hypothesis, they end up with the answer or the plausible target on this side, not the other side. But anyway, what we did is, since we were not sure how good this label-free analysis is, so we used, we purchased antibody to confirm the result. And then we realized, as many people pointing out right now, if you use immunoblood compared with the mass spectrometry-based method, they are different in one aspect. Let me see. So so in the mass spec, the ratio between these two cell lines cannot be very big. You always end up with something like the ratio is below 10. But using immunoblood, you can probably you will get a better, probably better or more accurate result. And once we realize, once we obtain the list, we can just use the traditional molecular biology technique. We can do a knockdown and or overexpression on the particular protein, and then we can see how they affect the cell migration and invasion. For example, like this one, we found FUCA2, this protein, they express more in the tumor cell, and when you use the concentration or the abundance of this protein on the cancerous tissue, you realize it's related to with the disease-free survival. So it's related to the patient's prognosis. So which is very useful for the doctor, because when the doctor performs the surgery, they can obtain the cancerous tissue and perform a immunoassay on this protein, and then they can predict the prognosis. Another protein is also interesting is actinin-4, using the knockdown and you can see the migration and invasion, they change. One thing is interesting is actinin-4 also has an effect on the cell morphology. For the when we treat the actinin-4 siRNA, so they will transiently knock down the abundance of actinin-4. Actually, the cell show different cell morphology. Usually the CL1-5 cell, they will show a spindle-like cell morphology. This it's close to the mesenchymal like cell morphology. So they are kind of carried through the so-called EMT, epithelial mesenchymal transition. It's not very clear here, but if you blow this up, you will find only these CL1-5 cells form these spindle-like cell morphology. If you knock the actinin-4 down, they are gone. So somehow this could be related to the cancer cell metastasis. So you can stain this protein using tissue microarray, collect the specimen from the patients, and then try to see if they are good for any purpose of diagnosis or prognosis. That's the result. For example, it's a diagnostic marker for the, if you compare the cancer cell to cancer tissue to normal tissue, or from patient to healthy controls, they all show very good differential power with pretty good area under curve values. Since we have so many, we get many potential targets. Actually, if you don't be picky about the criteria, the list is about 100 proteins. I would mention it a little bit later because recently we started doing using a mass spec method called multiple reaction monitoring. We can verify the target, many targets at the same time. Okay, so like this one, COL6A1, the knockdown and overexpression shows this protein is related to the migration and invasion. And the other two proteins, and pretty similar result on the clinical assay. And then we start thinking about among these targets, maybe some of the targets, they are somehow related. For example, maybe one protein is regulating another protein. So we show some examples. This is one. A1AT is in the list. Fibronectin is also in the list. After talking to my colleague, he's an expert in fibronectin. He told me when lung cancer cell, sometimes lung cancer cell, they will have fibronectin polymerized on the surface of the cell. Then they can attach to the distant site. So that's interesting. And then we find out that A1AT is actually require, shouldn't say that. A1AT can actually promote fibronectin polymerization by using the tail vein injection. So what we did is we inject the metastatic cell and also the transient knockdowns for the A1AT. You will find, so for the CL15 cell, you can see the fibronectin, they wrap around the cell on the surface. But when you knock it down, they are gone. They are not they don't polymerize on the surface anymore. If you inject these two cells into the tail of the mouse, then they get into the circulation, and then they might colonize on the lung to form the tumor nodule. And then you can just count the size, count the number of nodules or the size. So what we have here is, yeah, I think that's the weight. Okay. So for the CL1-5 cell, either treat with scramble control. That's what the data shown here. Always Liao, you will get pretty much the similar result. And you compare them with the CL1 cell treated with the A1AT knockdown, and you will see the ability of the cells to form the nodule is related to the A1AT abundance, and also to the fibronectin polymerization. And recently, we started working on the, as you can see, after we get the list of candidates from the proteomic, or we call this secretome analysis, then we can just verify the target one by one. Recently, mass spectrometrists, they developed a assay. They can work on the multiple target at a time. So actually, it's a very old mass spectrometry technique. For example, I can draw my blood and monitor 10, 20, even 30 drug target and their metabolite, their concentration in the blood. So, but they are for small molecules. But people just use it for the proteins. But you can not just do this on the protein because the size is big. So what we do is we digest the proteins from the blood into small peptides and then apply the similar procedures to it. So what we did is we get 100 proteins in the list, and then we can just do a lot of, for example, we call it the qualification process. If you cannot measure in the plasma, then you don't need to worry about it. You will never be able to find an answer for those things, and then you can just make this list shorter, and then you compare these proteins, and then we end up with 9 proteins with statistically significant result. So these are 3 examples. For these 3 proteins, they have enough diagnostic power shown by the ROC curve. I'd like to thank my group members, especially for my PhD students, to complete this work, and the funding resources. We have a Ministry of Science and Technology in Taiwan. They provide major funding. They also fund a national research program for genomic medicine and biopharmaceutical, and my university, and also the Ministry of Education. I'd like to thank 2 of my colleagues. Professor Su, he actually provides all the specimens because he's a doctor in our medical center. And Professor Chen, he's an expert, I mentioned to you, on the fibronectin polymerization on the cell surface. Thank you so much for your attention.
System for the cell culture. Usually the hollow fiber system, we use it to mimic the pharmacokinetics of drugs, right? Because we have an input, we have also an output, so you can culture the cells in a dynamic system. Mm-hmm. So the idea behind is, are you exposing your cells to drugs and then trying to look at the secretome from the medium, or okay. How are you implementing that in the
okay.
The procedure of getting the secretome?
Okay. Let me make sure I understand your question.
Yes.
Hollow fiber has been used for the dynamics of the. I think that's like a neurotransmitter or thing.
Yes.
Yeah, because I'm no expert about the hollow fiber, but I do know colleagues they are using hollow fiber. Yes. They stick the hollow fiber into the brain of the rat or mice. And then they can continuously sample the fluid from their brain or body fluid. Mm-hmm. Now, in that way, I think that's what you refer to the kinetics, because you can monitor a lot of metabolite or neurotransmitter concentration along the time. Okay. That's the only thing I knew about the hollow fiber. Okay. But I learned this is because I try to find a culturing system. You can culture a lot of cells. So we use different systems. I can't remember all of them, but they are different kind of culturing systems. Give you increase the surface, so those cells can attach to. And one day I found this system. It's called FiberCell. It's a company. Actually, it's a very small company. 15 years ago. 2004, no, 12 years ago. So I called them. Actually, they sent someone to my lab while I was visiting UCLA. So it was a very small company. they use this system to produce antibodies. Antibodies, they are secreted protein, right? So you can just culture the cells and harvest the antibody all the time. I explained to them I want to collect secreted protein from mammalian cells, so I just purchased this cartridge and brought them back to Taiwan, and my students work out all the data. Yeah.
Thank you.
Yeah. So so basically these cells just grown between the fibers. These fibers, I think you can think they are just like a capillary in your tissue. Right? So very good nutrient and gas and waste exchange, mass transfer between the cells and the reservoir. Yeah. And there's a pumping system. There's a pump, small pump, just like your heart. So these cells I think they are more happy. But if you grow them in a monolayer, in a dish,
They confluence, and then at some point
yeah,
you cannot grow more.
I think they are very stressful.
Yes.
These cells. Yeah. Yeah.
Is there a reason why you're not using more quantitative techniques and also you can multiplex more samples? So here you're doing label-free of just 2 cells.
Are you referring to I use label-free method? Yeah. The main reason to use label-free system is it's cheap. Okay, we don't get too much funding in Taiwan. So at first we used the isotope label tag, but that's too expensive for me. So we switched to the label-free method. I think, pardon?
What method do you use?
You know what method we use? So for mass spectrometry, you always get a signal.
Right.
But it's difficult to do the absolute quantitation because you have to calibrate somehow. But we don't even do the absolute quantitation here. We just try to compare the signal abundance between two cell lines. So what we did, you mean what kind of software we use?
Like in label free, you have different methods of doing label free. Like what's the secret label free?
Oh, you mean like a spectral count and okay, we just pick the signal, and we did not use any software before. We just pick the signal and quantize them and do it by manually or do a little bit simple programming using Visual Basic. That's what we did. Now we use a commercial software called Proteomics. I think it is the very good label-free software product in the market because they actually work.
You mentioned when you use this system, you got really huge biomass net compared to the literature, right?
Yeah, that's true.
So how large is this filter?
Oh, okay. It's like the size of my thumb. It's a little bit smaller, and it's like 10 centimeters long. So it's a very small cartridge.
So in this case, can I use this system to get like a biomass? Especially I'm working with like inside protein screen, not outside proteins. So can I use this system?
Oh, I see.
Large biomass?
Yeah. If you want to grow large number of the cells, and these cells are attached cells, all you need is a large surface area. There are many kinds you can get. But for the hollow fiber, you not only you get a large surface, they grow into three-dimensional structure and very good circulation. I think that's a difference. But if you just want to get, also this kind of setup, it's so easy to collect the secreted proteins. That's why I use this system. If you just want to use a cartridge system to collect the proteins in the cell, I think you can use this one or other system. It could be cheaper. This one is not cheap.
So is it commercially available?
Yeah,
How much is it?
probably more than one manufacturer right now. But the manufacturer I contact with it's called Fiber Cell. Fiber Cell, C-E-L-L. Yeah, you can just Google. It's easy to find.
But if you're interested in getting cells, this is not an ideal system because
Yeah.
So he's collecting the secretome. So if you want to harvest the cells, it's a more complicated system because now you have to kill the cells and they are all in there. You need some sort of a fermentation or cell suspension system with a spinner flask and things like that. It also depends on whether you need millions of cells, billions of cells, or trillions of cells. Right? And that scale will. But if you just need billions of cells, I think there's so many other methods.
Yeah.
Harvesting protein from here from the cells will become a problem.
Yeah, that's true.
So very nice talk. So we've done similar work. One of the complications that we weren't aware of when we did secretome analysis at that time was exosomes, especially when we did functional analysis. So we switched pretty much using FBS that has been depleted of exosomes. So that might be something that you keep in mind for further research, especially if you're using a highly concentrated mix of spent media for cell culture. There's going to be tons of exosomes in there if they're out there.
Okay, thank you. Actually, when we submit our first manuscript, the reviewer already pointed out what you are analyzing. So we have to think carefully. So basically, we are analyzing traditional secreted protein. For those proteins, we did an N-terminus sequence analysis. We found a portion of them, they have a secreted protein sequence. And we also, maybe through many other non-classical pathways, exosome, definitely they are one of them. We are also analyzing, like a CLIC1 I just showed you, they are not secreted protein, they are not even in the exosome, they are cell surface proteins. But sometimes they shade. When they shade a small, small segment of the protein into the conditioned medium, we still get the sequence, amino acids, we still get, using mass spec, still get the target. So there are many possibilities about them. But what, for me, what I can say is, if they are useful for biomarker or not necessarily the mechanism.
Yeah, I agree.
Yeah.
We might have the same reviewer.
Yeah. So exosome is definitely one of them. Sorry, I didn't get into the detail about what kind of proteins it could be many different mechanisms. We don't even know. But what we know is if you can find it from this model system, likely you will find this protein inside the body. So that's what we are looking at. Thank you for your information.
Thank you. Thank you very much.